Hi, my name is Sam, and today I will be presenting the project Identifying Cellular Cancer Mechanisms through Pathway-Driven Data Integration. So about this project, we all know that cancer is a genetic disease where accumulated mutations of genes induce a reprogramming of existing cellular pathways. Current approaches typically identify cancer pathways as those most internally perturbed by cancer-driven changes in gene expression. However, when we have a closer look at the known cancer drivers, we find that they perform hub roles between pathways. So our hypothesis is that in order to identify cancer pathways, we shouldn't be looking at those most internally perturbed, but we should be looking at those that have the biggest changes in their pathway-pathway interactions. So we suggest our pathway-driven NMTF to learn an embedding space that captures the organization of pathways in the healthy cell. Then, once we have this embedding space, we define two condition-specific pathway and gene embeddings. So diseased embeddings and healthy embeddings for both pathways and genes. Then, based on these embeddings, we can compute two heuristics in this space. Our NMTF centrality, which measures the functional importance of a given pathway or gene, and our moving distance, which for a given pathway or gene measures the functional disruption of its relationships within the cell in cancer. We apply both heuristics then to go on and predict cancer pathways and the genes that are involved in 4 major cancers. These are our data. As you can see, for each of our 4 cancers, we collect a case and a control network. So to do this, we take the protein-protein interaction data from BioGrid and the gene expression data from Protein Atlas for these 4 types of cancer and their corresponding control tissue. Then for each of these networks, we take the subgraph that is induced by their expressed genes. Additionally, we take curated pathways from Reactome, so the Reactome pathway ontology, and for each tissue, we only take those pathways that once we induce them on either the case or control network, they induce a subgraph that's at least 10 and up to 100 nodes. So now for our PNMTF model. As you can see in the data model below, our goal is to learn a latent pathway representation UP for every pathway in our set of curated pathways. Then, on the other hand, we also want to learn an embedding space V that organizes these latent representations. To do so, we first compute for every pathway in our set of curated pathways the matrix HP, which encodes how that given pathway interacts with the rest of the genes in the cell. For a given pathway P, we compute HP as the submatrix that is induced by the genes in that pathway on one axis and the rest of the genes in the cell on the other axis. Having defined our PNMTF model, we can go on to define pathway and gene embeddings. So for a given pathway P, we interpret multiplying its latent pathway representation matrix UP times SP as projecting that pathway in the embedding space V. Each row of that matrix corresponds to the embedding vector of a given gene that participates in that pathway in the common space V. We then represent that pathway as the average of its gene embedding vectors. To also get a diseased representation of a given pathway or gene, we simply just fix our space VT, which we learned based on the control PPI network, and solve our PNMTF model again, but this time based on the case, so the cancerous PPI network, and compute our embeddings again for pathways and genes. Having defined our healthy and diseased pathway and gene embeddings, we can now define the following 2 heuristics. First of all, NMTF centrality, which we use to measure the importance or the functional importance of a given pathway or gene, which we compute as the Euclidean norm of its embedding. Then our moving distance, which measures the disruption of a pathway or gene's functional relationships, which we define as distance, the Manhattan distance between its healthy and its diseased embedding. And now for our results. Our first step is obviously to validate our model. So here we are showing that PNMTF actually manages to capture the organization of pathways in the cell. To do so, in this case for the lung control PPI network, we embed all the pathways in the shared space V, applying hierarchical clustering, and extract clusters of pathways. We show that these clusters of pathways are much better separable than if you were to use a standard NMTF model that doesn't include reactome pathway information. And the clusters of pathways that you get are also much more functionally coherent. So having shown that PNMTF captures the organization of the cell, we move on to use our NMTF scores to predict cancer pathways. So in this case, we take the top 100 highest scoring pathways for our centrality heuristic, our moving distance heuristic, and our hybrid heuristic, which is the average of the former two, to predict cancer pathways. We find that we outperform two methods that are part of the state-of-the-art, which is classic gene enrichment analysis and Anubix, which is a crosstalk enrichment analysis method. Having shown that we can predict cancer-related pathways, we move on to predict cancer-related genes within those pathways. So within the set of top 100 highest scoring pathways, we predict 100 top scoring genes as being cancer related for each of our NMTF heuristics. Again, we see that we outperform the state of the art. In this case, it's the classical eigencentrality, which predicts cancer related genes based on their importance, their topological importance in the PPI network. Finally, we perform a case study in lung cancer, where we try and validate the top 50 predicted genes for lung cancer and the pathways that are involved. So in this case, for the genes that are predicted for lung cancer, we can validate 49 out of the 50 genes predicted to be involved in lung cancer, and spurred on by this high validation rate, we are quite confident that our only remaining prediction, SLC47A1, for its role in the SLC-mediated transmembrane transport pathway, is highly likely to actually be cancer-related. So in conclusion, I have shown that PNMTF captures the organization of pathways in the cell, and it allows you to predict cancer pathways while simultaneously also providing insight at the gene level. Thank you for your attention, and I hope to see you in the poster room. Bye.